All right, we'll get back to it. We have one more presentation on bacteria to get through of other important bugs to know. And then we will go into some viruses for our last hour of the day today. So we've discussed our gram-positive rods. Now we're going to go into our gram-negative rods. So these are gram-negative bacteria that are rod-shaped. Again, we can't have spores if it's a gram-negative bacteria. So there's no spores that are gram-negative. So we're just dealing with the world of gram-negative bacilli, gram-negative rods at this point. We're going to talk about some that are aerobic versus anaerobic. So we will start there. So our aerobic, meaning oxygen-loving, gram-negative, non-enteric, meaning they're not in the stomach, bacilli, okay? This is a large group of non-spore-forming bacteria. They say non-enteric, so that's hilarious, but they could. There's some that live in the intestines, so it's like not fully true. But really, this group, because they're aerobic, they're typically not seen in the gut. Zoonotic, meaning they have different vectors. They like to live in the respiratory system, soil, water, etc. Most of these are not medically important. So we're only going to be looking at the medically important ones that are true pathogens or opportunistic pathogens. All of them have this outer membrane lipopolysaccharide of the cell wall. So this endotoxin that we talked about at the beginning. So the ones that are probably the most important, Pseudomonas really is an important opportunistic pathogen. Bordetella and Legionella are the main human pathogens. So we're going to really focus on Pseudomonas, Bordetella, and Legionella in this category. These are kind of a flow sheet to help you differentiate things. So if this helps you, great. Gram-negative rods, these are aerobes that do not ferment carbohydrates. So Pseudomonas, Bordetella, they fall in these categories. These are facultative anaerobes that ferment carbohydrates. So these ones like lactose, E. coli, Klebsiella, Citrobacter, and Enterobacter. Non-lactose fermenting, we can see a whole host that we're going to get into, really Salmonella, Shigella proteus, Serratia yersinia, and then Haemophilus and Pasteurella will be our last kind of two that we'll look at. If you are like me and like tables, this is broken down kind of like we had for our gram-positives. This shows the organism, the condition based off of organ system that it could potentially cause. So you'll see some of them are much less common when it comes to pathogens versus some are very specific, like Bordetella pertussis, whooping cough, right? Has one pathology really to note, versus others can cause a lot. Klebsiella, wound infections, meningitis, pneumonia, UTIs. So you can see there's some variation. And then we have this separation between E. coli, as in seen commonly in a UTI, versus E. coli, the hemorrhagic form, the O157H7 form, which is associated with hemolytic uremic syndrome and acute diarrhea that's bloody in nature. Again, continuation of this table. So if all you can do at this point is look at bacteria names and pathologies, this table would be a good place to start. So getting back to our bacteria types. So Pseudomonas, this is a gram-negative rod. It is motile. It can live outside on its free living. So it can live in the soil, water. It can colonize plants and animals. We will see it as a contaminant in homes and in the clinic. It uses aerobic respiration. So it doesn't ferment carbohydrates. It actually uses oxygen. So it produces oxidase and catalase then. We can see the specific species, Pseudomonas aeruginosa. This specific species can live in the intestine in 10% of normal people, so it can be opportunistic. It's resistant to a lot of things. We see it often in contaminants of stuff that we find in the hospital. So we often associate this as an opportunistic pathogen with hospital-based infection. It can cause issues with burns. It can cause issues in patients that are potentially immunocompromised. Oftentimes when I've seen this clinically, pneumonia and UTIs is what we commonly will associate with it. but abscesses, otitis, endocarditis, the list goes on and on. An interesting component of this bacteria is it does have a sweet smelling odor, and it does look green to blue if you grow it out. So those are some distinctive characteristics of Pseudomonas aeruginosa. But think of it as an opportunistic pathogen associated with a hospital stay or an immunocompromised host. It can cause all kinds of different infection types. We see it more commonly in pneumonia and UTI, but no, if it is some type of infection and you're speaking about someone who's in a hospital or who's immunocompromised, it could be Pseudomonas as an answer. All right. Some other gram-negative aerobic rods include Burkholderia, Actinobacter, Strenophomous. I would not worry about any of these three. The only gram-negative aerobic rod I would know about is Pseudomonas and the species aeruginosa. Okay, so we're going to skip these because they're not important. Main reservoir for Pseudomonas, soil and water. Okay, moving to Brucella, brucellosis. Not important. We're going to skip it. Okay, there we go. Bordetella pertussis. This would be our next important gram-negative rod. It's encapsulated, so it can evade the immune system. It causes pertussis or whooping cough. We commonly see this more in children. It's an acute respiratory syndrome. It can often be severe and life-threatening. You can have healthy carriers. That's your reservoir. And it's transmission via direct contact or aerosols, so droplets. Virulence factors, it has receptors, the bacteria that bind to cilia, the actual respiratory cells that have cilia on them. So that's where it can bind and start to grow and infect. It also has toxins that can destroy and dislodge those cells. So that's where we can see the cough, where we may potentially have harder clearance of mucosa and other things, because the cilia typically is partially why we're able to clear some of that stuff out of the respiratory tract. So you get a buildup of mucus, you can have blockage of the airways. We do have a vaccine, our DTaP vaccine, which has a toxoid. It's a toxoid-based vaccine. That's your big piece with Bordetella. Kids, respiratory infection, paralyzes or destroys the ciliated cells that line the respiratory tract, that pseudostratified ciliated epithelium, and gram-negative, facultative, aerobe, broad. Those would be the big things with Bordetella pertussis. Okay. Ecalogenes, opportunistic. I wouldn't worry about it. Legionella. This one's important. Legionella pneumophilia. Legionella is in water. It hangs out as a close association with amoebas. It can cause pneumonia and Legionnaire's disease. It's the one that's the most severe that we hear about. Legionnaire's disease we've seen in males over 50 or in elderly patients. We see symptoms of fever, cough, diarrhea, abdominal pain, and the pneumonia fatality rate is 30 to 30% only in Legionnaire's disease. There are treatments for antibiotics, but Legionnaire's, I would think about gram-negative, broad, in water a lot, associated with amoebas, is potentially a vector, can cause pneumonia, can cause Legionnaire's disease, which is the most severe form, more common in patients who are elderly. Okay. All right, now we get into the good stuff. This is the, did they ever ask the difference between DTaP and Tdap? Yes, on NPLEX 2, no on NPLEX 1. Good question, Hannah. Any questions about vaccines? We're only going to come up with how the immune system responds to the vaccine. So what is the immune response? They're not going to ask how to give the vaccine, why to give the vaccine, to whom should have the vaccine. Those are all NPLEX 2 questions, treatment-based, but good question. Yeah. All right, so our Enterobacteriaceae family, entero meaning gut. So these are going to be our gut-based bacteria. They're all gram-negative. They're non-spore forming. They're all rods. Lots of them live in the soil, water, decaying matter. They also could be in the large bowel of animals. The most frequent cause of diarrhea is through these enterotoxins from those enterobacteria. We'll see a large amount of infections associated with these. The most common opportunists, because they can live also healthy in our normal flora, is E. coli, Klebsiella, Enterobacter, Serratia, Protis, and Citrobacter. So these are the 6 you should definitely know. of our Enterobacteriaceae. Some key characteristics: they can ferment carbohydrates, they are oxidase negative, they're anaerobes, meaning they don't like oxygen. There's flagella in most of them, but not all. And there's two kind of forms that we talk about. There's the coliforms and the non-coliforms. This just depends on if they ferment lactose or not. For purposes of this exam, probably not helpful unless if your brain needs more categories to organize these bacteria, these bugs. This is what like a dichotomous key would look like. I don't feel that this would be important for the purposes of your class or this NPLEX exam. There's virulence factors that can be common to most of these bacteria. These are just going to cause pathogenicity and cause certain immune responses. The H surface antigen is the one that has flagella. So if they have the H antigen, they're motile. The K antigen is a capsule or fimbria. So they're going to be able to evade the immune system and attach to mucous membranes. The O antigen is the somatic or cell wall antigen, meaning that their cell wall has extra protection against getting bored through by like a natural killer cell or an ST cytotoxic cell. It also is the antigen that's associated with endotoxic shock, the LPS component that can actually cause a lot of the pathogenicity, the pathosymptoms. All of them have the O toxin, the endotoxic shock components, that antigen. Not all of them have the H or the K antigen. All of these, the pathogenesis is really associated with their endo and exotoxins. So we're going to talk about first E. coli. This is our most prevalent enteric bacillus. It's the most common aerobic and non-fastidious bacterium in the gut. It is aerobic and anaerobic, so it can do both. It is a facultative. There are some that have virulence by plasmid transfer. There's others that are opportunistic. The pathogenic strains of E. coli, we can group them in some categories. Our E. hec is our enterohemorrhagic. These are the ones that are going to cause hemorrhage and bleeding and kidney damage. Our enterotoxigenic, our E. tech, is going to be our most severe diarrhea due to heat-labeled toxins and heat-stable toxins. This also has fimbriae, so able to adhere to mucous membranes. We have our EIEC, enteroinvasive, which is going to cause more inflammatory disease of the large intestine. Our EPEC, which is like linking to more of a wasting form of infantile diarrhea. The enteroaggrative, EEC, the cause of pediatric diarrhea. And the DAEC, totally different, diffusely adherent, which is a minor cause of pediatric diarrhea. Which ones should you absolutely know? Know EHEC and ETEC. Know these two kinds. EHEC because it's the hemorrhagic form. ETEC because it's the one that has the most common toxin forms that we see. So know these two forms of E. coli as pathotypes. Otherwise, understand just E. coli as a general pathogen. We'll look a little bit further into this. The majority of clinical disease is transmitted among humans. Pathologic strains, we can see infantile diarrhea is the greatest cause of mortality among babies. It's 70% cause of traveler's diarrhea, and it's about 50 to 80% cause of a traditional UTI. So it's commonly seen in conditions that aren't deadly, but it's a common pathogen that we will see and encounter. The role of E. coli in food infections. So O157H7, it's the somatic O type. It has that O antigen. It also has that flagellar H type antigen. So that's the O and the H that we're seeing here. It's the antigens that this form of E. coli has. This is the enterohemorrhagic strain that's involved with infections from fast food hamburgers, a reservoir in the cattle intestine. It is also known for its ability to fuse due to its cell wall receptor with the host cell membrane, creating a port directly to where the bacteria can secrete toxins directly into our cells. Not super great. The toxin then enters the host cell, binds to our ribosomes, and stops or disrupts protein synthesis, leading to programmed cell death and shedding of your intestinal mucosa. And that's where you can get some of that bleeding, that hemorrhage, and then obviously destruction or decreased absorption. So this would be, if you had to know one severe type of E. coli, this would be the one to know. We test meat now to make sure that we don't spread this, which is good. Okay. Those are your E. coli's. UTIs, gut infection, no EHEC, ETEC, and your most famous kind, O157H7, known for its O antigen and its H antigen. It's motile, meaning it can move, and it can fuse with the cell membrane of our host cells, sending its enterotoxins directly into our cell, disrupting and killing them and sloughing them off. Okay. Other bacteria kind of in a category that were related, other coliforms, these were lactose fermenting bacteria. Most of these can infect almost any organ. They're opportunistic. They can be drug resistant. So Klebsiella pneumoniae normally lives in our respiratory tract, has a very large capsule, but in someone who's immunocompromised could cause pneumonia, meningitis, bacteremia, wound infections, and UTIs. I would say pneumonia and UTIs are the commonly associated things I see with a Klebsiella infection for someone who's immunocompromised. So this would be a question where you have a patient who has some immunocompromised state, maybe cancer, maybe HIV, maybe even diabetes. You could see multitudes of ways they could present an immunocompromised patient who develops an infection, either a UTI, pneumonia, or otherwise. And if they're describing to you a gram-negative rod, think Klebsiella as a possible etiology. Enterobacter is typically more seen in UTIs. Again, same, opportunistic, not typically pathogenic. Citrobacter, opportunistic UTI or bacteremia. And then Serratia, more seen in burn or wound infections. Again, opportunistic. It also is bright red when you grow it out. It's red pigment. It's very beautiful, actually. Okay. Some lactose negative enteric bacteria include Proteus, would be probably our one opportunist that we want to know about, or our true pathogens, Salmonella and Shigella, which we'll look at. So Proteus and its relatives are typically found in soil, manure, sewage of polluted water. Proteus will swarm on the surface. It likes water. It's involved in UTI infections, wound infections, pneumonia, septicemia, infant diarrhea. Proteus also tends to be a infection where you'll see a more pH alkaline environment in the urine. So if someone has a Proteus-based UTI, the urine will tend to be more alkaline in nature instead of acidic in nature, and that can be a sign. I don't think they'll ask you that question because it's more diagnostic. So I'd say that more on NPLEX 2. But that is an association with that Proteus-based infection, Proteus-based UTI. Your Salmonella and Shigella, Salmonellosis and Shigellosis, some GI involvement in diarrhea. They can also affect other syndromes or symptoms. They're always pathogenic. They're never opportunistic. Salmonella has two species. Salmonella enterica is the one that we see potentially infecting humans. Obviously, Salmonella is commonly seen in gastroenteritis and food poisoning. We also can see it as typhoid fever. So the formal kind of designation of Salmonella and its serotype, it could be, it depends on kind of its subspecies name. You don't need to worry about those pieces. So the Salmonella Typhi, this is our typhoid fever etiology. We see it entering in with ingestion of food or water, contaminated typically by feces, so that fecal-oral spread. You can have an asymptomatic carrier. You could shed some bacilli from your gallbladder, but where the bacilli likes to live is they adhere to the small intestine and cause that invasive diarrhea that we associate. There are some vaccines available. Okay. Shigella and bacillary dysentery. Shigellosis is common incapacitating dysentery. It's due to Shiga toxin. We'll see fever, nausea, severe stomach cramps, and watery stool with mucus and blood. Some of the etiological agents, you can see there's lots of different Shigella types, subtypes present. Typically, they won't care about the subtype. They'll just list Shigella. It invades your villus of your large intestine. It doesn't perforate the intestine. It doesn't typically enter into the blood. It can enter into Peyer's patches, though, that immune pathway, that immune system in your digestive tract, and will instigate a pretty severe inflammatory response due to its endo and exotoxins. Yersinia is probably not going to be tested on, but could You see it in wild animals, fish, fruits, veggies, water. And then the one that will be tested on potentially is plague, Yersinia pestis, and the plague, the black plague. I've only seen this really tested because of its vector. So remember, it's spread via fleas on mice. So that's where you get the mice piece. The vector is a flea. And that's the plague. Bubonic plague, flea bite, lymph, necrosis, and swelling of lymph nodes. That's the bubo. That's the only time I've seen Yersinia on NPLEX is when talking about bubonic plague and asking for that vector component. Maybe they'd ask about bubo, but I think that's too easy. They'd probably ask you about the vector, wanting you to say like rat or mouse, but in reality, it's the flea that bites you that's actually causing the spread. The flea is just hijack on typically a rodent. Okay. Zoonotic diseases, typically we don't worry about them as much because they're not as common to be passed over to humans. We do see Haemophilus influenza, though, as one of our etiologies for meningitis. So we will look at that. And then Pasteurella, because it can provide some opportunistic infections as well. Pasteurella typically is associated with people who are living with domesticated animals or close human contact. Animal bites and scratches can spread Pasteurella. It's more at risk for people who are immunocompromised. Haemophilus is named for the fact that it loves blood. So Haemophilus, think of like heme, think of blood loving. They're very small gram-negative rods. They're sensitive to drying, temp extremes, disinfectants, so they're pretty easy to kill. They require heme, NAD or NADPH, so they like our blood because there's plenty of that supply. You can see some of them colonize the upper respiratory tract or vagina, but really the one I want you to think about is H-flu for childhood meningitis. That's really what I want you to think about when you think about the Haemophilus genus. So H-flu, acute bacterial meningitis, is going to be your big kind of association here. You could potentially see an epiglottitis, but I think bacterial meningitis, and you're looking at your categories, your young children really being your main category, or obviously immunocompromised elderly adults. Haemophilus ducreyi, I mean, there's a chancroid STD association. I doubt they would ask you about this. I really feel H-flu is the one I've seen the most. Okay. Great. Flying. Any bacteria questions before we move into our discussion on viruses? With where you're at now in your study, my recommendation is to take the bacteria that we talked about today that are the most important, create a list with one disease that's associated with each bacteria, and one or two either virulence factors or micro identification techniques. That could be morphology and gram, that'd be fine. Or if you already have that in your brain, it could be morphology, gram, and something else. Maybe one of its exotoxins or virulence factors of some kind, or how it evades the immune system. That would help you plenty. If you have the bacteria, it's disease associated, and two things that make that bacteria unique. That'll be probably enough for most of your bacteria and microbe questions. There will always be some specific kind of funky ones on NPLEX that you've maybe never heard of before. That's fine, right? That's any section, you're gonna see that. So that's really where I'd sit with bacteria at this point in time. Okay, all right. So the virus discussion, I want to spend this next half an hour going through kind of some generalities about viruses, and then we'll spend the last hour looking at some specific viruses and comparing and contrasting. The same is true for viruses at this point. We're going to talk about some understanding about viruses, but I would go virus, that's common, disease associated, and DNA RNA. That's what I would go with for now, this close to the NPLEX exam. If you can at least know that, that's often how they'll ask the question. They'll want to know the viral association with disease, and then they'll often give a second, right? A second option, trying to get you to either choose DNA or RNA or double-stranded or single-stranded. Those are the common secondary associations they give you with viruses. So some general properties of a virus, I'll let you read through the table, but this just talks about kind of generalities. General structure, remember, a virus has to use cell machinery to survive. It's not surviving on its own. It's not having a vector. It can't live as a fomite. It needs our own machinery, our cellular machinery to live and to be able to replicate. And viruses are small in relationship to bacteria. These are bacteria. These are our viruses. So remember, these are very, very small, tiny structures in comparison to a single-celled organism of our bacteria. So when we look at viruses as a whole, we'll see that they can have a covering that's either capsid or envelope. Envelopes aren't found in all viruses. And they'll also have a central core that has their nucleus, so nucleic acid stuff, either DNA or RNA, as well as some proteins or enzymes to help with division. When we look at the general structure of viruses, we can look at capsids. All viruses will have a capsid. This is their protein coat. This protects their nucleic acid. This protects their DNA stuff or their RNA stuff. The capsid is made from identical units called capsomeres that are made of protein. The capsid plus the nucleic acid are called the nucleocapsid or nucleus capsid. Some viruses then have a secondary covering to protect them called an envelope. Ones that don't are naked. We call them naked viruses. So this would be an example of a naked virus with the capsid covering its nucleic acid component versus a virus that has an envelope, a capsid, and its nucleic acid component. Often on these envelopes, we'll see different proteins, like a spike protein, an M protein. These can be proteins the immune system uses to identify and to create antibodies towards. When it comes to structural types of viruses, I'm only giving you this stuff because sometimes these terms will come up in NPLEX. I want you to have name recognition, so if you see them, it doesn't freeze you. The two structural types is helical and icosahedral. Helical is what it sounds like. It's a helix. It's cylindrical nucleocapsid versus icosahedral. It's typically multi-sided, has 12 different corners. There's some variation in this structure. Some of these can be enveloped. This would be a helical example versus an icosahedral. I mean, this is kind of an example. It doesn't look that great. This would be a better example of your icosahedral here. When it comes to the envelope, typically viruses that can infect humans or animals are ones that have envelopes or structures, but not all of them. This envelope is acquired when the virus leaves the host cell. Typically, it sometimes can even take part of the host cell's cell membrane as part of its envelope, or can take some proteins from the cell as part of its envelope. But the exposed proteins on the outside of its envelope are called spikes, and they help with adherence. Why is the capsid or envelope important? For protection. Okay. There are a few viruses that are atypical, that are complex. Pox viruses in particular lack a typical capsid and instead have a lipoprotein or lipoprotein layer that covers it. Some bacteriophages also have a polyhedral nucleocapsid as well as a helical tail, as well as attachment fibers. So we'll look at bacteriophages as they have a different structure. Bacteriophages here, you can see you have your capsid head, this collar, this sheath, the tail pins, and almost what looks like spider legs, tail fibers. Essentially, think about bacteriophages as viruses that could inject viral DNA into another cell type, typically often a bacteria. This is how certain bacteria like Shigella got their Shiga toxin, was thanks to a Shigella bacteriophage. So oftentimes, if you have some of these toxins that a bacteria can secrete, that is thanks to a bacteriophage for that bacteria, a virus that gave that bacteria some specialized DNA or RNA component. So now that bacteria has an additional toxigenic or additional virulence factor. But bacteriophages typically are viruses that infect other bacteria or other single-celled organisms. Okay. We're actually using these medically now in some instances to treat diseases. Dr. What's her name? Oh my gosh, it's escaping me. It'll come to me on the break, but there's an ND, she's using a bacteriophage to treat some like really significant diseases in Portland. It's pretty cool. I don't know if that's officially FDA approved, but it's fine. Anyways, nucleic acids. Okay, so the viral genome is either going to be DNA or RNA. It's never going to be both. It's only going to be one or another. This is important to note because it's going to tell us what type of cellular machinery is needed if the virus has to go all the way into the nucleus of the cell, or if it can replicate in the cytoplasm using translation, right? If it's RNA, that's already what we need to be able to make proteins, right? If it's DNA, it's going to have to go into the nucleus, get converted, get replicated, then get transcribed, then get translated, right? So it's going to have to take more steps if it's DNA. Versus if it's RNA, it can just go get translated into a protein right off the bat. So we'll see, depending on the viral genome or nucleic acid, it's going to depend on how it's affecting the cell it invades. DNA viruses are typically double-stranded. So if you don't know, guess double-stranded, but some are single-stranded. Okay, so if you can know, great. If you don't know, assume double-stranded because the majority are, but some aren't. RNA viruses are typically single-stranded, but some are double-stranded. So a good rule of thumb is if you have no idea about the genome of your virus, or if you know the genome type, but you don't know if it's double or single, if it's DNA, choose double. If it's RNA, choose single. You will get some of them wrong, but you'll get the majority of them right. Okay. Typically, RNA viruses, they're segmented into separate RNA pieces. ssRNA genomes are ready for immediate translation. We call those positive sense RNA. So if the virus itself has an ssRNA genome that can just go straight into translation in the cell, we call that positive sense. If we have an RNA genome that has to get converted into an appropriate form first, we call that negative sense, because we're having to work backwards, the virus is, to actually be able to replicate its proteins. Okay. There are sometimes preformed enzymes like polymerases, replicases, reverse transcriptases. These are things obviously that can help with replication. The virus's whole goal of getting inside a cell is to replicate, right? That's its whole purpose in life, is to do that. And so if it has some enzymes to help it do that process, fantastic. That means that process can go forward easier. So there's lots of different classifications. I'm going to really lean into our DNA and RNA. There's many different ways that virals multiply. So the general phases in animal virus multiplication, we have to have some type of absorption. So the virus has to bind to the cell surface, and it has to actually then get through via penetration. That's how our genome enters the host cell. Then there needs to be some sort of uncoating where the coat, that capsid, is removed, exposing the nucleic acid. Then synthesis, creation of our viral components, assembly, reformation of the viral subunit together, and then release, getting it out of the cell, either via some type of exocytosis or often apoptosis or hemolysis, destruction of said cell lysis. And so this is, if you're visual, this would be what it looks like. Not all host cells, or not all viruses like every host cell. So some viruses like specific types of cells that are going to host them. That will depend on the virus, that will depend on the cell, that'll depend on receptors, right? So if the virus collides with a susceptible host cell, a host cell the virus can actually bind to and absorb through, then it will bind to its receptors on the surface and it'll absorb. If it doesn't bump into a cell that it's able to actually bind to and penetrate, then viral infection, viral replication will cease to exist. It will not take place. The virus cannot replicate outside of a cell. Some viruses can really only infect specific types of cells, like hepatitis are hepatocytes, or human liver cells. Polio are intestinal and nerve cells. Rabies, tons of different cell types, right? Of a variety of mammals. So it depends on the virus for what types of cells it has, antigens that can bind to receptors, or receptors that can bind to antigens. Okay, and we can see that example here. When it comes to penetration, we see kind of two different ways that viruses can penetrate. Endocytosis, where it's engulfed and taken in via a vesicle or a vacuole, or fusion, where the envelope of the virus actually merges directly with the membrane, resulting in the nucleocapsid's entry. Kind of it's actually pushed into the cytoplasm because it's membrane fused. You can see either or, it depends on the structure of said virus and this host cell. So this would be endocytosis. versus actual host cell membrane binding and then it being engulfed and taken into the membrane itself and released. Depending on whether the virus is DNA or RNA, it's going to depend on its replication process. DNA viruses are replicated and assembled in the nucleus. RNA viruses are replicated and assembled in the cytoplasm, as a general rule of thumb. Positive sense RNA can go directly into translation into proteins. Negative sense RNA has to be converted into positive sense message first. The assembled virus will then bud or lyse the cell to leave. And it's budding. I don't have a picture of lysis. The host cell damage varies depending on the virus, depending on the replication, depending on if the virus is in a latent or active stage. But any of these effects, we call them cytopathic damage to the cell via the virus. So it could change the host cell size and shape. It can include cytoplasmic inclusion bodies from actually bringing the viruses into the cell, nuclear inclusion bodies from obviously having them inside the nucleus, cells fusing to form multinucleated cells, cell lysis, actual destruction, DNA alteration, or even transformation of cells into cancerous cells, depending on how much they hijacked that replication process. So lots of damage can take place. You also can have persistent infections where cells that harbor the virus aren't immediately destroyed. This especially rings true for viruses that don't cause cell lysis, instead do endocytosis or exocytosis. This could be weeks or lifelong. And you can also have viruses that will go inactive and become active again. That's how you get that chronic latency state. Measles can stay hidden in brain cells for multiple years. Herpes simplex, lifelong, right? Once you're infected, you never fully clear. It's always going to live out in some cells in your body in a latent state. Herpes zoster, chickenpox and shingles, another example. You get chickenpox as a child, it'll go away but remain latent and could reactivate as shingles in your later years. So you can see potentially different viruses have this different latency timeline, different latency period. That's why we can get chronic infection. We talked a little bit about cancer and tumors. Any virus that can initiate a tumor, we call an oncovirus. So we think of HPV, a papillomavirus, cervical cancer, or Epstein-Barr, Burkitt's lymphoma. These are the two that we probably are going to most commonly associate with those cancer-causing viruses. There's many others, but these are the two that really come to mind first. I mentioned bacteriophages. Bacteriophages are the bacterial viruses, phage. So these are viruses that are going to typically inject some type of DNA, RNA into a already formed bacteria. We've looked at E. coli a lot, but there's many bacteria that have a bacteriophage associated. We would call a bacteriophage an E. coli bacterial virus or an E. coli bacteriophage, meaning that's a bacteriophage that's associated with E. coli bacteria specifically. So these are the 6 stages in phage replication. I kind of already mentioned those already, but this is what it looks like. And again, it looks a little bit different because we're going into a single-celled organism that can replicate on its own. We'll see that there's the bacterial DNA in a capsid, and the viral DNA is being almost like injected like a syringe. From there, you'll see replication of the phage components, assembly of new virions, maturation, and then lysis. And it does kill off the bacteria. But if those bacteria were able to divide during this process and some DNA was incorporated into the bacteria, that DNA will be incorporated moving forward. And so then that bacteria will now have mutated or altered moving forward into the future with a new virulence factor. One thing I wanted to mention in this initial lecture is lysogeny. This is our silent virus infection. This is essentially what happens if a phage or a virus doesn't complete the lytic cycle. So some DNA phages called temperate phages will undergo absorption and penetration, but they don't replicate. The viral genome instead inserts into the bacterial genome, and it will then become an inactive prophage. The cell does not become lysed, as we saw here, but rather the actual cell can continue to divide. The bacteria itself can continue to divide and grow. The prophage, the component that was inserted into the bacterial genome, is retained and copied And that's all happening over and over and over again in a phase called lysogeny, essentially a phase where it's not a lytic phase. It's not destroying the bacteria. The bacteria is able to divide and grow. You can have induction later on. So you later on, after that bacteria is grown and divided and done its thing, you could have induction of that component of the genome to create new bacteriophages. So later on, it can move into its lysogenic phase or its lytic phase, but it can start initially in a lysogeny or lysogenic phase where it's not killing its host cell. So this is an example of the lysogenic stage where things are just replicating, no one's dying, no cells are being lysed, versus its lysic stage or lytic stage where you're having the virions being released. Some phage genes that have been inserted in the bacterial chromosome. We see that in Corneobacterium diphtheriae, Vibrio cholera, and Clostridium botulinum. All of these have potentially undergone a phage gene that gives them more virulence factors and makes them more pathogenic to their host. All right. The other components that they could mention, although I doubt, would be prions, they're misfolded proteins, no nucleic acids. The only time I've seen these asked is in Creutzfeldt-Jakob syndrome or bovine spongiform encephalitis, mad cow disease. When I've seen this asked, it's essentially asking about what is this caused from, a bacteria, a virus, a prion, a parasite. So that's where I would lean into that piece. I wouldn't necessarily, or they're asking maybe about what a prion is. It doesn't have nucleic acid. It's not DNA. It's not RNA. It's literally a misfolded protein. It's a protein that's folded wrong, incorrectly. The other kind of virus-related type things, satellite viruses. These are viruses that need other viruses to replicate. Adeno-associated viruses replicate only in cells that have adenovirus. That would probably be the most common cause. The other one would be Hep D, hepatitis D. That hepatitis form needs cells that have been infected with hepatitis B to replicate. So those would be your two examples of a satellite virus. Viroids are just short pieces of RNA. None have been necessarily identified in humans. Okay, great. That was our generic virus lecture. So I want to break now because we're gonna go into the types of viruses that we should know for the last hour. So I want to take the next 10. So we'll come back at 10:53, and then we'll go to the end and we'll go through the important viruses to note, and then we'll be done. Take a break. 10 minutes. Oh, there we go.